Thanks very much, Sarah. And I just want to quickly get some notes up here for myself as well. Great. Hopefully you can all still see the main screen. It's really my pleasure to talk to you all today about these innovative DNA-based methods for biodiversity monitoring. So in the talk today, I'll cover some of the context around biodiversity monitoring in the mining sector specifically, introduce DNA-based methods, how they can be used, who we are, and what work we're doing with Anglo American to apply this technology globally for more informed decision-making. So just in terms of some context, and we've had some brilliant talks just to lead into this. The one from Andy just now really set the scene. But here we are with the World Economic Forum Risk Report of 2021. And once again, biodiversity loss is ranked as a top global risk in terms of likelihood and impact, so 5th and 4th place respectively. And while being down one spot from 2019 owing to COVID and infectious diseases, this is an increasing trend from previous years and will likely increase in the years to come. And interestingly, up the top right, there's global shapers' perceptions, and these are the young generation, the entrepreneurs, and their perceptions are that the environmental risks specifically have a far greater likelihood and impact compared to the views of the multi-stakeholders who actually were responsible for putting this report together. Fortunately, the increasing global appreciation of the importance of biodiversity loss and the environment, including the ecosystem services, has led to a greater number of mining companies and industries incorporating ESG+I frameworks and making stronger commitments to biodiversity targets like no net loss, net positive impact, nature positive, and ensuring that these are applied to the mitigation hierarchy, which you can see down here. And this is partly owing to greater pressure being placed on them through governments and regulators and financiers, and in line with the Equator Principles and the IFC performance standards, but also because of competitive behavior, public perception, and far greater scrutinization of supply chains by discerning customers, as we've heard. And I really believe the financiers and insurers have a really positive role to play in this space, as we heard on Monday from James Fryer, by offering finance and insurance options to projects that include only strong and clear biodiversity and ESG+I commitments. So over the past couple of years, reviews have shown that there's been a sharp uptake in mining companies' commitments to biodiversity, specifically from 2003 to 2016 and possibly beyond. We just don't have the latest data. But in 2014, a review was done by the IUCN Extractives panel. And at that point in time, all ICMM members had some form of commitment to biodiversity. And this was a 50% increase compared to 2003. So there you can see that rise over time. But alarmingly, over time, a number of companies have started to change their commitments. And as a result, those commitments have become unclear, or companies have started retracting their commitments altogether. And of course, this is reflected quite badly on many companies and was viewed as corporates backing away from public accountability for biodiversity impacts and dependencies. And this is concerning. But why was this the case? And I really liked Andy's talk because I think he set the scene here to say the model and what needs to happen. So some of the problems that lead to the retractions of biodiversity commitments or hesitancy to commit to them in the first place include limited corporate traction, difficulties with actually measuring the outcomes of those measures that are put in place, slow uptake from the finance industry to apply pressure, lack of objective repeatable data, and this is possibly one of the biggest issues, and we're going to touch on that in this talk. The difficulty to relate back to reference or baseline conditions and actually compare the new data that's being collected to the reference conditions. And then this need for the science-based criteria and metrics that both Andy mentioned. But also, as we know, biodiversity surveys historically and using conventional techniques are very costly and very time-consuming with long-term commitments needed. And unfortunately, during the time that these new commitments came out for NPI and NNL, so no net loss and net positive impact, there weren't a lot of pilot sites actually being studied and being looked at to help formulate those metrics and refine them. So with the problems and the challenges of biodiversity monitoring in mind, we're in a very fortunate position now with 21st century technology and recent scientific discoveries. And these have presented some fantastic opportunities to assist in this space. And DNA-based monitoring affords scalable, high-capacity biodiversity monitoring, really providing data now at unprecedented rates and also with relative ease. And this affords me the opportunity to introduce Naturemetrics. We were founded to bridge the gap between molecular scientists and environmental managers, and putting the tools in the hands of those who need them and making those tools accessible. What tended to happen is the molecular scientists were coming up with fantastic new discoveries and tools, but then publishing them in obscure journals, and this never made it into the hands of the environmental managers. Currently, we're based in the UK, but we have a global outlook, and we're working with clients from different sectors around the globe on projects ranging from research to compliance and statutory monitoring. And in effect, we do a full end-to-end service from provision of sampling kits through to the laboratory analysis, and then the computational processing and reporting. But I want to dive in quickly to what these DNA-based monitoring methods are. So metabarcoding is an approach that can be applied to many different types of samples. This could be an insect sample from a malaise trap or a pitfall trap. It could be a soil or sediment sample, or it could be a water sample either from the ocean or from water bodies nearby. Essentially anything that contains the DNA of multiple different species. We extract the total DNA from the sample and we sequence it on a high throughput sequencer to generate a profile of the taxonomic diversity contained in the sample. And in fact, we can process many different mixed species samples in parallel, generating vast data sets within a matter of weeks. Now, the beauty of eDNA and metabarcoding is the ease with which a sample can be collected. The kits that we provide contain everything you need and have been designed for ease of use. So even in remote sampling locations and specifically for the tropics. So using the example of a biodiversity survey of water, perhaps around a mine site, you can simply collect a water sample and process it in 15 minutes on site using our filters. And you don't need any cold storage or any means to reduce clogging, because that's already built in. So you don't need pumps, you don't need batteries, you don't need equipment. This is all nice and light, and you could put it in a backpack. What happens is the DNA then gets stuck on the filter, and you send that filter to the Naturemetrics lab, and then we process it. And in the lab, our expert team go through a series of steps. We extract the DNA, and then we can do this for a range of taxonomic groups, and this is where it really gets exciting. So we can even do multiple groups from a single sample, and normally there's some DNA left over, so we could go back and do this analysis and amplify the DNA for up to 4 different groups. Then what happens is we sequence that data, and we have about 100 samples being run normally at a time on the sequencer, and that spits out about 300,000 sequences per sample and a data file that has about 30 million sequences. And that's a heap of data. That's an immense amount of data that we've never been able to generate before. And what do you do with all this data? So the first step is we sequence that data, and then we start to match those sequences to the species that they belong to. And this is a sophisticated step, and our bioinformaticians have specially designed workflows to tackle this. It relies on having a reference database of DNA sequences for the region that you're interested in. So our team can then check the taxonomic gaps for your project and sites, and we can even do this before you begin. And then we can also advise on how to address them through barcoding campaigns, where you can start to generate the fingerprints for those reference libraries. But at the end of the day, you'll be presented with a scientific report detailing the species found at your project site. And now you've got something that looks recognizable as a species by site table. And everyone wants to know whether the numbers of sequences here have anything to do with the actual species abundance. And the answer is that for animals living in water like fish, it does indeed give a good indication of the relative abundance of different species at a site. So how can these immense data sets be used? Whether freshwater, terrestrial, or marine, onshore, offshore data obtained from DNA-based methods, whether it's eDNA or soil metabarcoding, can be used to inform all stages of the mitigation hierarchy. and asset life cycle from exploration all the way through to closure, avoidance through to offsetting. And using small organisms obtained from sediments or invertebrates, site restoration can be tracked in terms of efficiency over time, and even whole biomes can be assessed, and critical communities can be monitored in relation to impacts over the long term. So this takes a look at the communities rather than necessarily individual species. We can detect those individual species, but we're starting to move towards looking at whole communities and how whole communities are shifting. Ultimately, this information will help us to learn what good looks like, and we can start to apply machine learning and predictive modeling on these data sets with the big data that's provided to help us determine efficiently what condition the ecosystem is based on the biodiversity data. And this brings us to the work we're now doing with Anglo American. A new partnership will see DNA-based methods being applied at 12 pilot projects in 10 different countries, with those sites representing 10 different terrestrial ecoregions. And I'll briefly go through just a subset of these pilot projects to illustrate the ways in which the DNA-based tools are being applied for projects at different stages of their life cycle, and also with different priorities in terms of the mitigation hierarchy. And hopefully this will give you a flavor for how these tools could be applied perhaps at some of the mine sites you might be managing or involved with. So the first is really at the exploration stage. And we've heard that there needs to be more of an impetus placed on collecting biodiversity data early on. And the value here is that a geologist or a technician, somebody who's not biodiversity minded or ecologically minded, has the ability to take with them some kits and get data at the earliest stages of a project. This could be 15 minutes spent sampling a number of sites around the mine, and then using those aquatic eDNA sampling kits to obtain information on species that might be present. And these could be high-risk species, so they could be highly threatened or endangered species, but we'll also get the whole community information. And this is really useful for baselining and early risk mitigation. And so here really in the avoidance stage of that mitigation hierarchy. Flinging across now to a more seasonal environment, and also one where it's in the middle of a design and planning phase, you now have the ability to collect immense amount of data during the baselining stages before there's any construction or development, and really characterize the diversity that might be present at a site. So here, this is an opportunity from Finland, where we are involved in a high diversity bog and mire environment, which is close to a Natura 2000 reserve. And here the eDNA can be used to collect information on whole invertebrate communities, vertebrates, fish, native mussels, and even looking at some of the sediments to understand the bacteria, fungi, and metazoa, some of the small groups that up till now have largely been ignored, but really underpin ecosystems. Coming across to the UK, we've got a project that's now within the construction or development phase, looking at ways of avoidance and minimizing impacts. And this is an underground mine in a national park, and really with a wide range of DNA monitoring happening here on different communities, from rivers, ponds, wetlands, and also sediments. And looking at vertebrates, invertebrates, fish, and also diatoms, which are excellent integrators of water quality. But a real value add here is being able to engage with the national park and also citizen scientists and really get them involved in collecting accurate, objective data through citizen science. Coming across to a desert and xeric shrubland here in Southern Africa. This is a highly arid area, water scarce, and you might say, well, how do you use eDNA now from water bodies? And the fascinating thing here is there are ephemeral pan systems that are highly seasonal, but you can also get aquatic eDNA from artificial watering points within the landscape. And this can allow you to see what animals might be present in and around the mining area and in land management zones. But also, because it's an area of high insect diversity, there is insect sampling happening on site, and we can now ease the way in which those insect samples are processed, removing the need to some degree from the microscope and augmenting data sets by looking at that whole sample and metabarcoding it. And then, of course, with offset and restoration targets, we can start to look at sites that might be earmarked for offsetting or for restoration, and we can start to track those sites through time. And then this is the penultimate one, is a marine site here of Namibia. And just to illustrate the fact that the same methods can be applied in the deep sea environment. So looking at benthic marine sediments and looking specifically at impact and recovery over time, but using groups like bacteria and metazoa. And in this particular instance, there's a strong focus on building up the barcoding database and the reference library. There's some fantastic invertebrates that have already been collected, and now we can augment that data set and sequence it and get the individual sequences for those species so that we can better inform eDNA and DNA metabarcoding going forward. The last is to show that this can also happen at a landscape scale within catchments and whole catchments within an entire municipality. So this is a metallurgical coal in Australia. And the idea here is to look at linking in with the basin association and partnerships that are already monitoring sites and receiving water bodies all over Queensland, and tie into those and be able to augment their data sets with far more data, possibly for some of the groups that they are already using, but maybe some that they haven't engaged with. So vertebrates, invertebrates, and fish. And this now just allows me to end off with talking about some of these opportunities. I think the DNA-based monitoring methods really allow for accurate, repeatable, efficient sampling. It allows anybody to go and take a sample, and therefore democratizing the way in which biodiversity data is obtained. It allows for reduced costs and timeframes, specifically reduced health and safety risks, and it allows for early risk identification for projects. But the big data that's generated can really help augment conventional surveys, but also lead to machine learning and predictive modeling opportunities. So I think this allows for really realistic biodiversity monitoring and also learning within consortia. So there are consortiums, and we're engaging with the biodiversity consultancy and FFI and others to really understand what net positive impact metrics, how they need to be refined, what data can feed into them. And I think the predicts model that we've just heard about could be a fantastic one to apply in this space. And we can feed the data that's obtained from eDNA into those models. And we can also link it to some of the satellite technology that we heard earlier. So really generating some fantastic data products for sustainable mining going forward. There are opportunities here for skills transfer and upskilling to staff. So really feeding into the S and the G and also the I parts of this, because we can even involve native communities in the sampling and putting the kits in their hands. So yeah, great opportunities for community monitoring, co-engaged learning and outreach. And ultimately, we can also leave a lasting legacy by building up these reference libraries that are so desperately needed for certain areas, and that way contribute to science and research going forward. So thanks very much for listening. Really appreciate that, and I'm looking forward to any questions and discussion.
Fantastic. Thank you so much, Vere. A massive round of applause from all of us on the other side of our muted microphones. Every time I listen to you guys and your updates, I just want to give you a massive hug, because what you guys are doing is truly phenomenal. Also, a massive thanks for giving us a tour through Anglo-American. Having spent quite a lot of my early career in that company, it was really lovely to see some of those projects and operations and how they're getting on. We've got time for one very quick question, and actually that comes back to the Anglo side of things. So you mentioned the likes of Sakatti in Finland, and then the slightly more developed project here in the UK. Are you seeing any evidence so far where the company is making use of the data that you're collecting and then altering the mine design or the plans based on what you're finding, or is it still too early to tell?
Yeah, it's a great question. It is still too early, unfortunately. I mean, these projects, this is like hot off the press, being able to actually talk about the fact that we're now partnering and coming in. So this year we'll see a number of the monitoring ideas kicking off at these sites. So possibly in a year's time, I'll be able to report back in a better way about the data that's being obtained. I think, you know, it also depends on the stage in which the projects are in the EIA process. And Sakatti is really early on in the EIA process. Whereas Woodsmith is a little bit more developed and far advanced. So certainly for mitigation and management, ongoing management, yes, I think the data will certainly feed into that. And our idea is to work with the mines to understand the management interventions that they can apply and do that in a smart manner.
Great. And one sneaky question, if I can, this comes in from Amanda. This is a nasty one, by the way. She says, number one, how expensive is the product? But then number two, how do you deal with the risk to the mining company that they/you find something that then slows down the project?
Great questions. So the first, in answering the first one, it is, if you compare the cost of doing a biodiversity survey for a number of different taxonomic groups, the relative costs actually, it's cheaper to go the eDNA route because you can cover more organisms for less time in the field and less need for expertise in the field. So to give you a sense, I mean, it's around £275 per sample, and that would include a single group like vertebrates. So you can get an immense amount of data from a single sample collected in 15 minutes, where you would need normally a group of 4 specialists to collect that data. So that should give you a sense, but on that same sample, you can then add additional taxonomic groups for around £100 per additional group. So now you can get 4 or 5 different whole taxonomic groups from a single sample. So that gives you a sense of the costs. The key thing here is actually looking at the study design, the sampling design, and making sure that that is intelligent. And I'm sure Andy will agree with that, and some of the others. You want to plan that early on and make sure that you've got a good sampling design in place. In terms of the second part of the question, sorry, can you just remind me exactly what that was?
Yeah, how do you deal with the threats to mining companies that you might find something that then either stops or slows the project down?
Yes, good question. I'm very relieved to see that mining companies generally are being far more transparent about biodiversity data. I think before, that transparency may not have been there because of risks and financing and everything else. But I think now there's far greater pressure on mines being transparent with data. I think that there's a sensitivity towards organisms that might be poachable or have high value on black markets. That is where you then need to be very careful in the data that's shared. But ultimately, if it's a species of high conservation concern, you want to know about that early on. So the take-home message, I think, from us is that apply these tools in the earliest stage of a project before it hits the late stages of an EIA. Apply it in the exploration phase of the project.
Brilliant. Thank you very, very much, Vere. I could carry on asking you questions for hours, but we do have our wonderful next speaker who's all teed up and ready to go. So ladies and gentlemen, please put your hands together for the wonderful Vere and Naturemetrics. Thank you very much, mate. Great.